Hello. I'm here today to talk to you about the immune system and the various ways that it works to keep us healthy. This lecture, although quite long, is limited in scope. It's just about the actions of the immune system cells. We're not going to speak about what we can do as osteopaths or with nutrition or with lifestyle to support the immune system. We're not going to speak of the many interesting ways that the immune system works with the digestive system or the endocrine system or the nervous system. We're not going to speak of the many ways the immune system can go wrong. We're not talking about autoimmune disease when the immune system fails to distinguish between self and non-self. We're not talking about allergies when the immune system decides that harmless bit of pollen is a very dangerous invader. We're not speaking about mast cell activation syndrome, which is a new and very interesting area of study around chronic inflammation. We're not talking about people who are immunocompromised. We're not talking about people who have chronic infections like EBV, where our bodies keep fighting but never quite win. We're not in this lecture speaking of what happens when the immune system overreacts. We will talk about cytokines, but we are not talking about the cytokine storm. Okay. Rather, this lecture is designed as a prerequisite for understanding these many things: allergies and autoimmune and mast cell, cytokine storms, neuroendocrine immune systems. But here, I really want to look at the health of the immune system, the miracle of the immune system, and get a glimpse into some of the depth and complexity. So I've designed this as a basic review and overview. Some of the lecture may be pretty simple if you've studied it recently or keep up with developments. If you're not that familiar with the immune system, or if you've been in practice a while, this might be good review. I taught 30 years ago. I taught immunology for the ESO, and many of the things I'll be talking about today were not discovered or clearly understood at that time. So it's really an evolving field. I see this lecture as a preparation for looking at viruses and post-viral issues, which we're in the midst of right now. I've tried to organize it in a way that's accessible. There's definitely some repetition. There's a lot of things I'd like to have talked about that I haven't had time for, like the embryology and the development of the cells. But I hope that it's of some interest. In any case, I'm Orion Evans, and I hope you enjoy this. Thanks. Studying immunology, it's a bit like going down a lot of rabbit holes. Each piece can be studied in more and more complexity. So, for example, we're going to look today at T cells, and we'll look at three basic types of T cells, but there are, in fact, dozens of T cells. And on these T cells are markers called CD markers, cluster of differentiation, and we'll be talking about 3 or 4 of those, but in fact, there are over 300 of them. The other thing about immunology is there are lots of initials and lots of different names for the same thing. So IFN stands for interferon, IL stands for interleukin. A cytotoxic T cell can be called a cytotoxic T cell, or a CD8 cell, or a CDL, or 6 other different names. In some areas, perhaps I don't go into enough depth, and in other areas, I definitely have too much information. And there is some repetition in this lecture as well, so if you don't get it the first time around, we will be going over it again. I really see using this presentation as a reference tool to go back and review information or learn more details, or just try to get an overview as we go through the material. In the student notes, there is some extra information if you would like to look at that. Let's begin with immune system functions. The basic function is it distinguishes between our self and our non-self. It protects our self, and it deals with the non-self. It surveys the entire body. It also has a relationship with other systems in our body, particularly the endocrine and nervous system. A few features of the immune system. It has discrimination. Not only can it tell whether something is self or non-self, but it can tell whether it's a harmless non-self or a dangerous non-self. It's flexible. Our bodies are constantly changing. Perhaps yesterday we didn't have a tumor cell and today we do. Perhaps yesterday we didn't have an infection and today we do. It then decides an appropriate level of reaction and makes a memory for the next time around. There are three main parts of the immune system that we're going to talk about. The first is the leukocytes, or the white blood cells. This is a mobile system. These cells move around the body and are the basis of our defense. The next part is the cytokines. Cytokines are proteins designed to communicate. They're made primarily by white blood cells. They have many different names. A lymphokine is a cytokine made by a lymphocyte. A chemokine is a cytokine which is chemotaxic. It draws other white blood cells to the area. An interleukin is a cytokine made by one blood cell to act on another white blood cell. A monokine is a cytokine made by monocytes. You get the idea. These are basically all proteins produced by the cell in response to a stimulus that binds onto the cell membrane of another cell and has a messenger or communication or regulatory function. There are over 150 of these, and we are not going to talk about them in specific details. But their basic function is to interact with and communicate between cells, particularly between cells of the immune system. They can do this in an autocrine fashion, that they secrete a cytokine that acts back on itself, or a paracrine function that it acts on nearby cells, or an endocrine function. These cytokines can travel in the bloodstream to cells far away. And it's important to remember cytokines currently have a bad name because of the cytokine storm, but in fact they're a vital part of our immune system. They're pro-inflammatory or anti-inflammatory. They have a lot of different regulatory effects on the immune system. So we have the cells, we have the cytokines, and finally we have the complement proteins. These are proteins which are found in the blood. They're plasma proteins, and they complement the immune system. They help it. They help the innate immune system, and they help antibody function. And mostly they do that by this process called opsonization, which is a way of labeling or tagging a pathogen. Basically, by labeling or tagging it, it makes that pathogen easier for the immune system cells to find and act on, or for the antibodies to find and act on. So this is the order of the lecture. We're going to introduce the players, the various white blood cells. We're going to introduce the two main strategies that the immune system uses, which is innate immunity or acquired immunity. We're going to look at some of the related anatomy, and then we're going to look at the sequence of events. What happens when we have a pathogen? We're going to put the pieces together, and we will focus on the virus when we talk about a pathogen. So let's begin with white blood cells. Hematopoiesis is white blood cell development. Not going to talk about the embryology in this lecture. We'll begin with the pluripotent stem cells, also called HSC, which are stored in adult bone marrow. These stem cells are used to make white blood cells, red blood cells, and platelets. We're going to just be talking about the white blood cells. So this is a slide of the various ways that blood cells develop. There's a small number of the stem cells present throughout life, and they develop into various white blood cells depending on local signals, like from osteoblasts or cytokines coming into the bone marrow or into the lymph tissue. And here's a chart that I've made that makes it a little more simple. There are basically myeloid white blood cells and lymphocyte white blood cells. Here's another chart of the myeloid slide, which we're going to start with. And you can note, looking at various slides that I present or that you find elsewhere in the literature, that it's not always exactly clear how cells develop. For example, mast cells in some charts develop from the myeloid progenitor and in some straight from the stem cell. So let's begin by looking at the myeloid cells. And I'm just going to run through these briefly. You have a mast cell. The mast cell interacts with IgE antibodies and produces an inflammatory reaction. Histamine, heparin, other inflammatory molecules. You can see all the nice granules in the mast cell which contain the inflammatory molecules. This is a dendritic cell. It's supposed to look like a tree. Also called a DC or APC cell. The APC is antigen presenting cell. These cells are so important in the immune system. They eat the antigen, they eat the pathogen, they process it, and they put it on the surface of their cell membrane and present it to other immune system cells, particularly the T cells. So without the DC presentations, the T cells won't get activated. If the T cells won't get activated, as we'll see, there's pretty much no specific immune response. Next, we have a macrophage, also known as an antigen-presenting cell. Macrophages are fun. They eat foreign stuff. They eat antigens, and they present those antigens to immune cells, particularly T cells. They are phagocytes. They like to eat stuff. They start as monocytes and they become macrophages, and they're brought to sites of infection where they can live for months, eating debris, eating bacteria, and working as antigen-presenting cells, which we will talk about later. Then there's a whole series of cells that are called granulocytes. These are all phagocytes, although they sometimes have other actions as well. They're made in the bone marrow, they're brought into the blood for transportation, and they live in the tissues for a few days. Like the macrophages, they eat invaders, they eat dead tissues and foreign particles and anything with an antibody attached. The primary granulocyte is the neutrophil. This is, in fact, the cell that makes up about half or more of all your white blood cells. It has a strong phagocytosis action. It also recruits other immune system cells, and you can tell it has this multi-lobed nucleus. It likes to eat stuff. The eosinophil is important for fighting parasites and nematodes. It's part of allergic reactions as well. The nucleus has 2 lobes, and the cell body has hundreds of these microscopic granules. Basophils, part of the histamine release, allergic and inflammatory responses. Also, you can see these granules. So, moving on. Here we are. We've done the myelocytes, and now we're going to move on to the lymphocytes. We'll discuss some of them. There are many more that are being discovered and studied. The lymphocytes are all part of the adaptive or acquired immunity, except for the natural killer cells. These lymphocytes move from lymphoid tissue, like lymph nodes, to the bloodstream, into the tissues where they're needed, and then back to the blood, and they can circulate for months or years until they're needed. So we're going to begin with looking at the B cells. These make antibodies. They're called plasma cells when they're mature, and there are short-lived plasma cells which work on the current infection. There are long-lived plasma cells which are hanging out till next time, and there are also memory cells. Here's a nice picture of a plasma cell. The natural killers are lymphocytes that are part of the innate immune response. So they're not so selective about what they can kill. They bind to cells that are infected with tumors or viruses, and they secrete cytokines. They're constantly talking to other cells. They're looking for cells that don't look quite right, that don't have the right self receptors on them, and they kill them. And here is the natural killer cell releasing chemicals which are going to kill that cell. We will talk about it a little more later. And then there are the T cells. There are many, many different T cells. The ones that most people know and we'll talk about today are the helper and cytotoxic T cells. And T cells are they're like the orchestra conductors. So they are regulating and talking to everything that's going on in the immune system. We've met the players. Now let's look at the basic way the system works. The immune system, we know, recognizes the self and attacks the non-self, differentiates between okay non-self and not so okay or dangerous non-self, helps us to resist toxins and damaging organs, and has the ability to detect and destroy cells that are infected, particularly with viruses or tumor cells. There are 2 aspects to the immune system. There's the innate immunity and there's the acquired or adaptive immunity. The innate immunity is the first line of defense. It's always ready to go. It's not specific. It's a very generalized, quick, and consistent response. It recognizes molecular patterns that are associated with pathogens, and it attacks them. It doesn't have any memory. If that pathogen comes around again, it won't say, "Oh, I recognize you." It'll just say, "Oh, you're a bad guy. Let's do something." There are 3 aspects or ways that the immune system works with the innate immunity. There's the cells, there's the molecules, and there's other barriers to prevent infection. So let's start with those barriers. That's things like barrier tissues, like keratin on your skin, or tight junctions on your mucosal linings, the antimicrobial saliva that we produce, the good bacteria in our gut or the stomach acid. These are designed to prevent us from getting infected or to kill pathogens early on. Then there are a whole bunch of cells and the way that the cells react. Basically, cells recognize the pathogen, okay? And these are the cells we've talked about, the dendritic cells, the mast cells, the neutrophils, the macrophages, the natural killer cells. There's a specific molecular pattern called the PAMP on the pathogen, and the receptor on the immune system called the PRR recognizes it. You get a variety of responses. You get phagocytosis, you get inflammatory cytokine releases, you get chemokines, which attract other white blood cells, you get pathogens killed directly and infected cells killed directly, and you also have a very important aspect of the innate immunity response, which is that it activates the adaptive immunity. Molecules, we've begun to talk about these already, cytokines and complement. The cytokines signal the immune system and nearby tissues and release chemokines and pro-inflammatory mediators. And the complement is the plasma proteins that help or complement the actions of the immune system. They basically recognize a pathogen or an antigen or an antigen-antibody complex and help things along by releasing nasty chemicals or opsonization, that is labeling it, and resulting in what's called the membrane attack complex. We're going to go through this again. And it can help bind and cluster the pathogen. Here's a little picture of opsonization. So that funny looking thing on the top is the complement protein, and the tubular thing is the, say, a bacteria, and it's attaching to a macrophage. And because of the complement protein, the macrophage can help to recognize that it's a bad bacteria and it eats it up. So the immune system, innate aspect of the immune system, is a quick response with no memory. Okay, let's look at acquired immunity, also called specific immunity, also called adaptive immunity. This immune system aspect is specific and powerful, and it has a memory. It reacts to a specific antigen that has a unique marker, which is a protein or polysaccharide molecule. Basically, your T cells and your B cells wait in the lymphoid tissues in your lymph nodes or your Galt or Ralt systems. One antigen can activate millions of T and B lymphocytes. The B cells make antibodies in response to antigens, and the T cells orchestrate the response. They have a multitude of responses to the antigens. Let's look at the B cell. B cells are made in the fetus in the bone marrow. They move to the lymph tissues where they hang out and wait until they're activated. Now, the antigens are either brought directly to the B cells through the lymph tissues, they circulate through the lymph node, or they're eaten by macrophages and presented to to the B cells, the macrophages go to the lymph tissues and say, hey, look at this, I think you might be interested. In response to this, it makes antibodies, which are specific to that pathogen, to that antigen, which are released into the lymph and into the circulation and can travel through the circulation to where they're needed. Okay, so a B cell becomes a plasma cell. That's when it's grown up. It makes lots of antibodies, and it makes memory cells and long-lived plasma cells for future needs. There are 5 different classes of antibodies. You probably know this from your practice, and we're not going to go into the details. IgG is the primary thing we're concerned about, certainly with viral infections. The basis of all antibodies is this Y shape. It has 2 heavy chains, those are the long ones, and 2 light chains, those are the short ones. And they make a Y. The bottom of the Y is called the Fc, or constant part. It's unique to each class of antibody, and it can bind to a white blood cell, or it can bind to a complement protein. The top is varied. The Fab binds to a specific antigen. The Fab portion is unique for each pathogen. I thought you'd like another picture, a 3D picture of what these antibodies look like. Here's what they do, and we're going to review this later in the lecture. They bind to that pathogen or antigen. They do agglutination, where, for example, bacteria get clumped together. They do precipitation, where the antigen and antibody become too large and are insoluble and are of no use to anybody. They can do neutralization, where they cover the toxic site of the pathogen. Okay, so for example, they might cover the toxic site of the virus so the virus can't get into the host cell. They do complement fixation. Remember the complement proteins. The antigen and the antibody and the complement form this thing called the MAC, the membrane attack complex, which can lead to lysis, or death, of the infected cell. Lysis just means the cell membrane is ruptured. There's ADCC, which is not a rock band, antibody-dependent cellular toxicity, where the IgG antibody helps a natural killer cell in its actions. Opsonization, we've talked about. It labels the antigen so that the macrophages can find it easily. It helps phagocytosis in other ways, and it remembers for the next time around. Then we have the T cells, also known as cell-mediated immunity. The naive or inactive T cells hang out in the lymphoid tissue, and they wait for the antigen-presenting cell, usually a dendritic cell, to present the antigen. The T cells cannot see the antigen on its own. It cannot see a virus. It can only see a bit of a virus protein that's presented on the cell membrane of the dendritic cell. Okay, these cells are now called billboard cells. These antigen-presenting cells are like a billboard holding up the important sign, which is the antigen, the MHC receptor on the T cell, and the T cell says, oh, okay, I got it. Something bad. reproduces lots of cells, lots of different kinds of T cells. There are the helper T cells, which regulate all the immune functions. They secrete their own lymphokines. They promote the growth of B cells and T cells. They make new T cells of all various different kinds. They are essential, as we'll see later, for antibody production and activation of B cells. They activate the macrophages, and they live for a long time. Cytotoxic cells, also known as CD8 because of the CD8 marker on their membrane. They recognize the antigen proteins on the surface of an infected cell or a cancer cell or a transplant cell. And it kills the cell. Apostis is a word that means kills the cell. Punches a hole in the membrane and sends in granzymes, which are nasty chemicals, to kill the cell. We also have regulatory or suppressor T cells, which prevent autoimmune reactions and also help to end the immune response when it's no longer needed, and memory T cells, which are pre-programmed for the next infection to allow for a stronger and faster response. And lest we forget, there are many, many different kinds of T cells, and they're all being studied in depth. New ones are being found. The specific actions are of interest to very geeky immunologists. So we've looked at the players, the cells and the various molecules involved in the immune system, and we've looked at the basic way in which the immune system works with innate and acquired immunity. And let's have a brief look at the related anatomy. The lymphoid organs are places for the cells to wait for the action, particularly the T and B cells. They're hanging out there and they're waiting for the lymph to come in and show them things. And the lymph brings antigens and antigen-presenting cells. The lymph filters through the lymphoid organs, through the lymph node, and the T cells and the B cells that are waiting there check it out. They have a look, and if they like what they see, they say, oh, that doesn't look so good, and we're going to be activated. And remember, there is associated lymphoid tissue in many places in the body: the spleen, the tonsils, the respiratory system, the digestive system. So in the lymphoid tissue, there's concentrations of T and B cells lining the sinuses and the venules. The lymph brings the fluid for surveillance. The immune system cells have a look, and then the lymph goes back into the circulation, into the lymph and the high endothelial venules, and the activated immune system cells and the antibodies get into the circulation. Here are a couple of microscope pictures of these cells: macrophages, T cells, B cells in the lymph node. Here's a few more pictures. You can get an idea of the density of these immune system cells. And here we are, a nice picture of the lymph node. So in summary, the immune system is complicated and effective. There's many different cell types, different functions, different cytokines, different complement proteins, all with different functions. And the lymphatic circulation and the lymphoid tissues are an important circulation and distribution system so that the immune system cells can be activated and can get to the places in their body where they're needed. Now let's put all of these aspects of the immune system together and look at the immune system at work. We're going to specifically look at viral infection. This is a general look at viruses, not specific to coronavirus, but there are differences in the way that the immune system reacts to a virus, to a parasite or bacteria. Remember, we're not looking at allergies or autoimmune reactions. We're not looking at the ways that viruses can mutate and make this reaction much more difficult. We're not looking at immune deficiencies or interactions with the neurological and endocrine system at this time. Before we start looking at the viruses, I want to make a little side comment on the markers that we've been talking about, because we're going to talk about those in a little more detail. Cell membranes have markers that are proteins or glycoproteins or lipopolysaccharides. They are on the cell membrane, and there are many names of them. We've mentioned some of them. There's MHC, which is major histocompatibility complex. There's a PRR, which is the pathogen recognition receptor. There's a TCR, which is the T cell receptor. And the CD, which is a marker that identifies what kind of T cell it is. Viruses also have markers. In general, they're considered antigens. There's the PAMP, which we talked about earlier, which is that pathogens have a molecular pattern that even the innate immune system can recognize as being non-self. Viruses will look at the capsid proteins that can be specific to the virus, viral proteins that are bound in the membrane coat of the virus, receptor binding domain, which are the bits on the spike proteins that allow those spike proteins to connect with a host cell membrane and get into that cell. Okay, so let's look at what is a virus. Basically, it's a bit of genetic material, RNA or DNA. It's some kind of protein coat and some kind of envelope. Not all of the viruses have a protein coat or envelope. A question that's been asked in the study of viruses are they alive? And most immunologists don't consider viruses alive. It's not a cell. It can't reproduce. It can't respirate, make its own energy. It can't digest. It can't make new proteins. It can't do much of anything at all unless it's a host inside a host cell. All the proteins in covering the virus are made by the host. All the genetic material is made by the host. Now, it's very clever in the way it can get into the host and use that system. So, what is a virus? Basically, as I said, genetic material, RNA or DNA. It may have a capsid, which is a protein coat of these repeating subunits of proteins. And then it can have an envelope, which can look very different in very different viruses. And it can be a lipoprotein membrane with glycoproteins stuck in that. There may be a lining inside, there may be spike proteins on the outside. Here's a few different shapes of viruses. We recognize that one on the bottom left, which is a helical virus with a jumbled ball of yarn kind of genetic material. And there's the rod-like viruses, which have helical nucleic chains, and those lovely alien things called bacteriophages. And here is how they look under a microscope. Let's look at the basic steps of what happens when we interact with a virus. First of all, the pathogen has to get into the host. We breathe it in or we eat it. Once it's in the host, it has to get into the cell. Remember, a virus is basically harmless until it gets into the cell. When it's in the cell, it hijacks the cell functions in order to replicate. So it makes new proteins, it makes DNA or RNA, it assembles new viruses. Then it leaves the host cell, having made maybe tens of thousands of new viruses. And I will note there's lots of opportunity in this process for mutating. The viruses don't have the kind of editing protection that our own DNA does. It leaves the host cell, usually kills the host cell, but not always. And then the replicated viruses, all these copies, get into new cells. And then we cough, and those infections get into a new host. Now, usually the viruses are not lethal to human beings, except if they jump species like Ebola, or it's a brand new mutation, or the host has compromised immunity. So the analogy I like to use with my patients is it's like somebody breaking into your house. And they have a key. The key fits into the lock. Remember, cell membranes have these three-dimensional receptors, which are like the lock, and the virus has these proteins in its coat, which are like the key. So the virus gets in, the burglar puts the key into the lock and opens the door and gets into your house. But it can't do anything because it's wearing a very big, heavy winter coat. And so there's a valet in the house that says, "Please, sir, let me help you with your coat," and takes off the coat. Once the coat is off, that person gets to work in your house. It eats all the food in your fridge. It turns on all your lights. It goes into your bedroom and does all kinds of stuff and makes lots of new burglars. Then when it's done using your house, it leaves. Sometimes it leaves through the front door, and sometimes it just blows a hole in your whole house and goes. So let's look at these steps in a little more detail. First of all, it has to get into the host. The pathogen has to get into us. We know the various ways that can happen. It can happen through the GI tract. We can have contaminated food or water to get Hep A or gastroenteritis. We have certain defenses there. We have the saliva, we have our acid in our stomach, we have the GALT system just underneath the small intestine, we have the good bacteria. A pathogen can try getting in through your respiratory system. We breathe in water droplets or aerosolized viruses, or we touch contaminated objects and put our fingers in their noses. We have also defenses there. We have mucosal linings in our respiratory tract. We have cilia in our nasal passages. We have the RALT system. The skin, the mucosa, the genitourinary tracts are all other ways in which viruses and other pathogens can get in. Insect bites, sexual contact, cuts or abrasions, And again, we have defenses. We have the keratin in our skin. We have mucosal linings. And of course, we can have a pathogen enter our bodies directly through the blood, through transfusions or contaminated needles. Once it's in our bodies, it then has to get into the cell and get to work. In order to get into the cell, it has to get through the cell membrane. The cell membrane is designed to keep bad stuff out, but the virus has tricky ways to bypass that. Okay. Once it's in the cell, there are membranes around other parts of the cell, like the nucleus membrane and the endosomal membrane. So it also has to get through those membranes. There's a lot of specificity. Different viruses have specific ways to get through. There's a variety of ways, which we're not going to talk about in detail. But basically, the virus attaches to the cell membrane receptor and interacts with the host receptors in that cell membrane, okay? And then uses a variety of techniques like punching a hole or fusing with the membrane. Each virus is different, and immunologists haven't figured it out because they have to figure out each virus separately. Okay, once it's inside the cell, it still can't do too much until that coating protecting the genetic material is off. Once that's off, it hijacks the cell machinery to replicate. It sheds its envelope, it sheds its capsid coat either on the way into the cell or once it's inside the cell. And it uses the cytoplasm organelles, the cellular functioning. It alters the metabolic pathways. Okay, it uses the glucose. It uses the ATP. Some viruses get into the nucleus of the cell. This is particularly difficult. The nucleus has a membrane that's very selective, and it has to find a way to make a hole in that membrane and get into it. Once the virus is in the cell, it gets to work and basically makes lots of copies. Here's a picture, just not necessarily for understanding the details, but to look at how complicated it is for the virus to get through this nuclear pore complex, NPC, in the nuclear membrane. Once it's got new viruses, it has to leave. Okay, it usually kills the cell lysis on the left, or it has various ways to bud through the cell wall without killing the actual host. So here's a nice summary. The virus, the parent virus, attaches to the cell membrane, sneaks into the cell, gets into the nucleus, makes lots of copies, and then its progeny leaves the cell. So let's look at what our immune system does to combat this invasion by a viral pathogen. A lot of this is going to be review because we're going to be talking about the innate and acquired immunity reactions. There'll be a little bit more detail, and it will be a little more specific to viruses. We know that there is the first line of defense, the innate immune system. This is in the first few days of infection. It's very effective for slowing or even stopping the initial infection. It utilizes a variety of white blood cells and cytokines and complement proteins. It also is very important in alerting the adaptive immunity system. There's two parts of the innate immune system, or I should say two steps that the innate immune system goes through. The first one is immediate, and that's to do with the sentinel cells. The sentinel cells are waiting in your tissues. The literature is not always clear about which cells. This is an area that's being currently researched, but in general, we know there are natural killer cells, dendritic cells, tissue macrophages, mast cells, neutrophils that are all in the tissues. These are the cells that recognize the PAMP, recognize the pathogen-associated molecular pattern with the pathogen recognition receptor. And I'm sorry about the typo, it should be PRR, not PPR. So cytokines start to be produced. The defense system starts to get into action. And the sentinel cells bring more white blood cells and complement proteins into the area of infection. Natural killer cells. Okay, these are in the blood, in the tissues, in the secondary lymphoid organs, and we know that these can kill infected cells as well as producing cytokines like interferons and tumor necrosing factor. They also have an important role that's recently been discovered in interacting and supporting dendritic cells. So the NK and the DC cells have a mutual support system going. Basically, natural killer cells recognize that there are not enough MHC1 receptors on the cell. So MHC is part of our normal cells. If those numbers are down, or if they see something that they recognize as being bad, they will attack it. And here it is, killing the cell. It produces perforin, which makes a hole in the infected cell, and then through that hole, granzymes, these cytokines, get through into the cell and kill the cell. Dendritic cells are also sentinel cells. These are these tree-like cells. They also eat the antigens. They check out stuff in the blood and the tissues. They eat the antigen, they digest it, and they present it on their surface. Then they travel to the lymph nodes to visit the T cells. And here's a picture. Again, you don't need to necessarily know, we don't need to go into the details, but at the top you have the antigen that comes into the cell, gets processed, puts on the outside of the cell, and then the T cells say, oh, I know that this is not good. Right. The cytokines, interferons, interleukins, tumor necrosing factor, there's many different types of each of those. They are made by the immune cells and also sometimes by the infected cells. As we talked about, they have autocrine, paracrine, and endocrine actions. Particularly important in this early system, the chemokines bring more white blood cells. They send out a signal, say, hey, we're in trouble here, we need some help, come and visit. They also make these adhesion molecules that let the white blood cells stick to the area. They also make, and I think it's not on the slide, they make cytokines that make the blood vessels more leaky so that complement proteins and other white blood cells can get into the area. Pro-inflammatory cytokines, antiviral cytokines that interfere with viral replication in an infected cell and also protect other cells nearby from infection. These cytokines encourage natural killer cell activity, encourage macrophage activity, encourage DC actions, and promotes other cytokines. So here we are, the cytokines finding the neutrophils, the cytokines on the blood vessel endothelium. And here's a nice picture of lots of cytokines getting into action. You can look at this at your own leisure. So here we have it. The immune system has the sentinel cells and the cytokines as a first step in the innate immune system response. The second step is more leukocytes, more proteins. Okay? So the white blood cells move in from the circulation. They say, hey, we got a call, we're here. The monocytes grow up to be macrophages, and they start to eat pathogens and they eat the dead cells. The neutrophils also are phagocytes. They eat and kill pathogens, and then they die and have to be eaten by the macrophages. The complement proteins, which we've talked about, they leak into the tissues where they're needed. The blood vessels are leaky, and there's chemotaxic cytokines. They can directly inactivate the virus. They can bind and cluster the pathogen in a process called agglutination. The most important thing they do is this process we've talked about opsonizations. They bind with the virus antigen so that that antigen can be seen more easily by phagocytes. And later on in the acquired immune system, they will bind with the antigen and antibody complex to help with that. So, the innate immune system action summary. Remember, there are two parts of this. Part one is the sentinel cells, and part two is the other cells and proteins that have been brought to the system. But basically, what you have is a quick response which can destroy the virus, destroy cells infected by the virus, stop viral replication in the host cells, regulate the inflammatory response. You have many different cells and complement proteins and cytokines at work to help stop this infection. If it does that, homeostasis is restored. But the other thing it does is it slows the infection to allow time for the adaptive immune response and actually activates the immune response. Those complement proteins and the antigen presenting cells will get those adaptive immune responses going. So I kind of think of it a bit like you have a problem and you call customer service and you talk to somebody about your problem, and maybe they can help you, or maybe they can help you a little bit but not completely, and mostly you get slowed down until you can get to a manager. And the manager is the adaptive immune system, which is a powerful system, which we're going to review now. Let's look at the adaptive immunity in a little more detail now. Acquired immunity, specific immunity. This is a later response. It's more powerful and it's more specific. The adaptive system does depend on the innate immune system. Antigen presenting cells have to come to the lymph tissues and have a little conversation with those naive T and B cells in order to get them activated. There's two basic responses which we know. There's the response from the B cells, which is to make antibodies. The antibodies have many different kinds of actions that primarily attack free viruses. The T cells find the viruses in the cells and help B cells and orchestrate responses. And it's important to remember that T cells are blind when it comes to free viruses. They just don't notice them. Unless that virus is in a cell and presenting with an antigen on the surface, it will not be able to act. So let's look at that T cell activation. The antigen presenting cell, that dendritic cell, eats the virus at the infection site, digests it, finds a little protein, and presents an antigen peptide on the surface. It attaches to the MHC complex on the surface of the cell, goes off to the lymph node, meets a naive T cell. Once it's there, there are actually two different kinds of activations, or possibly three, that need to happen in order for that T cell to be activated. That MHC complex and the antigen on the antigen presenting cell meets the TCR receptor on the T cell. In addition, the CD site, CD28, meets another receptor called the B7 on the antigen presenting cell. When that happens, the T cells differentiate into a variety of effector T cells. They leave the lymph node into the afferent lymph, into the HEV, the high endothelial venules, to go to the target tissues. Here's another slide which I just like the picture of. In this left-hand picture, the orange and red are the TCR, that's the sites on the naive T cell. The blue is the MHC, except because it's immunology and everything has all kinds of different names, it's called the HLA site here. And that little green blob is the antigen, okay? And here you can, on the other side, on the right side, you can see a diagram of the MHC protein on the APC in green, the T cell receptor in black, and that little tiny foreign peptide in the middle, and then a couple of other of these receptors that have to be stimulated at the same time. And here's a much easier picture of what happens. The dendritic cell and the T cell say, let's have a little conversation. I have something you might like. And the T cell says, an antigen. I'm going to wake up. Okay, and as we talked about, there are lots of T cells, and we're going to just check out a few. Cytotoxic T cell, also known as CD8, CTL, TC, cytotoxic lymphocyte, or killer T cell. They are activated by the APC. They're helped by a variety of cytokines. They multiply extensively, tens of thousands of copies, into the lymph and bloodstream. Okay? When they find an infected cell, they recognize that antigen MHC complex on the surface of the cell, and they kill it. Okay? And here is a nice picture. You have the CD8 cell with its TCR receptor. You have the MHC receptor molecule on the infected cell. The killer cell makes a hole in the infected cell, sends some granzymes in it, and kills the cell. So it's very similar to what a natural killer cell does, only it's specific. And because everything in immunology is complicated, here's another picture. The picture on the left is that system we were talking about, the TCR-MHC complex. And on the right is a slightly different pathway that they've now found can also kill the cell. And let's just remember that the killer T cells are going to kill infected cells. Helper cells. Also CD4, Th1, Th2, Th, T helper, Th17. There's a whole bunch of them. They are activated, as we talked about, in the lymph. Okay? The MHC The TCR, you can see on that bottom right, TCR in blue, MHC in purple, and the antigen in green. And then it has some helper receptor sites on the cell membrane surfaces. These cells, once they're activated, move into the circulation. They activate the B cell, which we're going to look at in a minute. They coordinate responses. They regulate the inflammatory response. There's a whole bunch of different subsets. I'm not going to go into that much detail on these diagrams. They can help the actions of the CD8 cells. They can help the B cell antibody response. They have some direct antiviral effects themselves, and they really can help the leukocytes and the macrophages and the neutrophils. They orchestrate with this response, and they do create memory cells. And for today, the last one we're going to talk about is the regulatory T cell, also called Treg or TREG or suppressor T cell or CD25. This basically operates on a negative feedback loop to downregulate effector T cell production. So it slows the whole system down. It reduces the effectiveness of the antigen presenting cells. It produces immunosuppressant cytokines. It turns off the immune response, and it's very important for our autoimmune responses. So here's a little summary. You have an infection, the antigen presenting cell says, hey, have a look at this. T cells get activated and they get to work to orchestrate the immune response. Let's have a look at B cells. The B cells are waiting in the lymph node, in the germinal centers. Okay. The antigens come and visit the B cells. They filter directly in the lymph through the lymph nodes, or they're presented by antigen-presenting cells. They become activated and are now called plasma cells, which are either short-lived and make lots of antibodies, or long-lived to hang out and wait for the next infection or become memory cells. The thing about the B cell is it really depends on the T cell to become activated. Without that helper T cell, it can't finish up being activated and making antibodies. So to begin with, the virus is recognized by a B cell. Okay? Taken into the cell and processed. Okay. The virus peptides are brought by the B cell to the surface and bound to those MHC complexes. The MHC2 antigen complex is recognized by the helper T cell. Okay. And the helper T cell activates the B cell. The B cell becomes a plasma cell, multiplies, makes antibodies. And again, immunology is ever more complex. We can see here in the middle is that MHC2 antigen TCR complex, but there are these other connecting bits that have to be in place before the B cell is activated. We've looked at the antibody actions already, but we can just review them again. The antibodies bind to the antigen, okay, which is a particle of the virus, right? Remember, those top bits of the Y are the antigen binding sites, all right? Neutralization covers the toxic site and doesn't let the virus get into the cell. Agglutination, okay, clumps the virus so it can't get into the cell. Precipitation makes the virus too large and clumpy to get into the cell. opsonization. It tags it. The antigen and the antibody together are an easier target for phagocytes. Complement fixation we've talked about. The antigen gets together with the antibody, and that gets together with the complement protein, and all three of those make a complex that can attach to a cell membrane and make a hole in the cell membrane, so it can kill an infected cell. Antibodies also can help phagocytes work. They also have this action called ADCC, which we're going to look at in a moment. And they can hang out and prevent reinfection. They sit at the mucosal surfaces to prevent reinfection. This is ADCC, antibody-dependent cellular toxicity. Okay, this is where the antibody helps the natural killer cells. Okay, so natural killer cells are part of the innate immune system, but they get helped by the acquired immune system. Okay, the infected cell has an antigen on the surface. The antigen attaches to the antibody site on an IgG. Okay, so the infected cell now has an antigen-antibody complex on the cell membrane. The Fc receptors on a natural killer cell, remember that's the constant part of the antibody, the natural killer cell has a site that recognizes it and binds it. So now you have the FAB of the antibody attaching to the cell membrane antigen, and the Fc attaching to the natural killer cell. Natural killer cell says, I know what to do. I'm going to kill this thing. So we have B cells that make antibodies that deal with the virus primarily outside of the cell. You have T cells that only see the virus when they're presented on the outside of a cell, an antigen presenting cell or an infected cell. It has to recognize an MHC antigen complex. Adaptive immunity. The antigens arrive in the lymph tissues either free or on antigen presenting cells. T cells and B cells are activated, multiply, get to work. Antibodies and T cells move to the site of infection. The T cells regulates and orchestrates the entire response. Somewhere along the line, memory cells are made, and somewhere along the line, the immune reaction stops. How does that happen? Nobody really knows. Okay, there's a lot of leukocytes that need to be cleaned up, but the memory cells need to stay alive. You want to avoid chronic inflammation. You want to avoid an autoimmune system reaction. You want to free up tissue space. You want to free up nutrients for other needs. The immune system is a very highly metabolic process. And how the whole thing ends, people really don't know yet. Certainly there are less antigens because the immune system is killing them. There may be a lack of nutrients because the system just simply can't keep up with the demand. There are regulatory T cells that slow the system down. There are anti-inflammatory cytokines. The T cells and other white blood cells just die and need to be cleaned up. So we don't really know why some cells become memory T and B cells and others are active and then die. We do know that the long-lived plasma cells produce low-level antibodies for a certain amount of time, as we're seeing in the current crisis. It's not always for a long amount of time. But the memory cells, the memory B cells and T cells are ready to be activated for the next time around. So finally, we are at the end. The immune system reacts to pathogens. It has an innate quick response. It has an adaptive, more powerful response. It resolves the issue and it leaves us with a memory ready to be active should we be reinfected. Okay, I hope this is of some help to you and enjoy it.